here we are at part 2 here on page 2. So you should have already thought about this warm-up, which is going to get you thinking about today's lesson. Again, you can read through the why, but basically these are two phrases that people like to use all the time when they say something is accurate or something is precise. And I would like you to actually be able to use those words correctly, okay? And then that's that target for today. So if you're going to say accurate or precise, do you really mean accurate or precise and when you say, okay? So let's get into this. So with accuracy and precision, we need to make sure we definitely have some definitions down first that you can use to make sure you're talking about these correctly, okay? So accuracy itself is going to be how correct you are to what the known value is, okay? So we know that water boils at 100 degrees Celsius. If you're measuring this in lab, are you getting 100 degrees Celsius exactly, okay? So correctness, okay? Basically, how close is it to the true value? Okay, like a dozen is 12, unless we're talking about a baker's dozen. If we're talking about a regular dozen, you're looking for 12. So when you're measuring something, you need to get 12 exactly. Okay. Precision, on the other hand, is different. So precision, or if you are precise, that's how close your values are to each other. So if you're measuring the boiling point of water, you're going to be accurate if you're getting 100 degrees Celsius. You're going to be precise if you measure it the first time you get 99, the second you get 99, the third you get 100, the fourth you get 98. Those are pretty precise because they're really close to each other. An example that wouldn't be precise is if you're measuring again the boiling point of water and you get 72, 155, and then you get 98. Those are so far from each other that they're nowhere near each other. Okay? So close to one another. Because as we take measurements in lab, you're going to be doing more than one trial in order to get better results, and you're seeing how close your trials are. So what I have next here is that data can be precise and accurate. It can be precise only, it can be accurate only, okay? Which that one generally comes into place when people are talking about taking an average of something, okay? And precise, neither accurate or precise. So you could be nowhere. You could just be all over the place. So let's look at this first task. We want to know what each picture is showing. So if you're throwing darts and you're aiming for that bullseye, and this is example A. So you threw all these purple darts, okay? We're trying to decide, are you accurate, are you precise, are you one of them or neither of them? So if we look at this one, the darts are nowhere close to the bullseye. Hopefully you would agree, okay? You're shooting for right here. So they're not accurate because they're not hitting the bullseye. I would say they're not precise too because they're all over the place. It's not like they're all together in the same spot, okay? So from that first one, I'm going to write here N, meaning neither. Okay, neither accurate or precise. This next one here, you're shooting for the bullseye, okay? That would make you accurate. There's nowhere close to the bullseye, so I'd say they're not accurate here. But look, they're consistently bad. They're throwing them in the exact same spot every time. That's nowhere near where they should be, but they're close to each other, which makes them precise. So P for precise. This one, they're shooting for the target in the middle, the bullseye. So there it is. They're really close to it every time, which makes them accurate. And they're also really close to each other. So all of the shots are really good. So I would say this is both because it's accurate because they're all right near the middle. Okay. Precise because they're close to each other in how they shot. Now let's try with some data in lab. Okay. So if you ran your first experiment, we're looking at the first column, then the second column, and the third column. Okay, because these are the different trials. You're trying to measure the boiling point of methanol. So instead of water, we're just looking at methanol. You know that the boiling point of methanol is 64.7 degrees Celsius. You could have Googled it. You could have looked it up in a book. You run 3 experiments, 3 trials for each. So we need to know if each experiment is accurate, precise, both, or neither. So let's look at this first one. They got 75.3, 43.2, and 87.4. I would argue those are not accurate because they should be getting 64.7. They're getting 75, 43, and 87. Okay, so not accurate. Now, I would argue that they're not precise either because they got in the 70s and then in the 40s and then almost 90. So they're not close to each other. So I would say N here for neither. Okay. I'm looking at experiment 2 next. So 65.3, 64.2, 64.9. These are all pretty darn close to each other. They're right around 65. Okay, so that's precise because you're close to each other. But the true value is 64.7, and they're getting right around 65, so that's also really close to that true value of 64.7, Okay, if you average these, I bet you'd be pretty close to that 64.7 number. So this one I would say is both, because they're close to each other and they're close to that true value of 64. Experiment number 3, 98.5, 97.9, 98.2. They're all in the high 90s, okay? So they're all pretty close to each other. That makes them precise, but they're consistently terrible because you're looking for 64, you're in the high 90s. So you're not close to that true value. So you're not accurate, but you're precise because you're close to each other, okay? So whatever was going wrong every single time, which means you close to each other, okay? All right. Now, you're going to practice this, and then we're going to do a mastery check on it as well. But if you look at this, you're, for the first one, looking at Lisa's values, all the way down to Lisa. Then you're looking at John's values. Then you're looking at Sarah's values and doing the same kind of thing that we did here. Okay, so how was Lisa? Was she accurate, precise, both or neither? Same thing with John. Same thing with Sarah's column. Okay, and then you'll answer this question for number 2. Then we'll check and make sure you got this before you move on.